Hi everyone, welcome back to my channel. In the previous video, I discussed regarding sickle cell anemia and its inheritance. In continuation with that, in this video, I'm going to talk about two problems related to sickle cell anemia. You all know that sickle cell anemia is a blood disease in which that individual will be having defective hemoglobin, due to which the RBC of that sickle cell anemia patient will be sickle shaped instead of normal biconcave disc shaped. Because of this sickle shaped RBC, that RBC will carry only very little amount of oxygen, or oxygen carrying capacity of that RBC is reduced, due to which that particular individual will suffer from severe anemia or lethal anemia. You all know that the gene responsible for sickle cell anemia is denoted by HBS. HBS, yes, refers for sickle cell anemia, and this gene is present on the chromosome number 11. So, the sickle cell anemia is a autosome linked trait. Autosome linked means gene responsible for sickle cell anemia is present on the autosomes, not on the sex chromosome. So, it is a autosome linked recessive trait. Recessive trait means only in the recessive condition the symptoms of sickle cell anemia appears in that particular patient or individual. So, when both the homologous chromosome of chromosome 11, if it has this abnormal gene or gene for sickle cell anemia, only in that condition that patient will be sickle cell anemia. Whereas in the case of heterozygous condition means one of the chromosome out of homologous pair having the normal gene or dominant gene, whereas another chromosome have this abnormal gene or gene for sickle cell anemia. In this condition, that individual will be phenotypically normal, or he or she does not show any symptoms of sickle cell anemia, but they are carrier. Carrier means they carry that particular abnormal gene or gene for sickle cell anemia for the next generation. So, though they are phenotypically normal, they carry that abnormal gene for the next generation. So, that is regarding the two conditions in which this abnormal gene or gene for sickle cell anemia can be present in the individual. We'll discuss about the first problem related to sickle cell anemia, and the first problem is, if a man who is carrier for sickle cell anemia marries a carrier woman, what are the chances of this couple having an affected offspring? So in this, a man who is carrier means he will be having HBA and HBS. So one of the homologous chromosome have this abnormal gene. So this carrier man marries a carrier woman. Again, the woman is also carrier, means she carry one of the abnormal gene or one of the chromosome out of homologous pair carry this abnormal gene. So, carrier man marries a carrier woman. So, obviously the genotype of the carrier man is HBA, HBS, and genotype of the carrier woman is HBA, HBS. These are the possible gametes produced by this parent. So 50% of the sperm produced by this man having the normal gene, whereas another 50% of the gametes will have this abnormal gene or gene for sickle cell anemia. Similarly, 50% of the egg produced will be normal, having normal gene, whereas the 50% of the egg produced will be having this abnormal gene or gene for sickle cell anemia. When this gamete having the normal gene or sperm having the normal gene combines with this egg having the normal gene, then it produces normal individual, and the genotype is HBA, HBA. Similarly, when this sperm having the normal gene combines with this egg having the abnormal gene or gene for sickle cell anemia, in that condition it produces carrier with the genotype HBA, HBS. When this sperm having the abnormal gene combines with the egg having the normal gene, then it produces carrier with the genotype HBA, HBS. Similarly, when this sperm carrying the abnormal gene combines with this egg carrying the abnormal gene, it produces sickle cell anemia individual with the genotype HBS, HBS. So, out of 4 individual, 1 individual will be normal, means 25% of the individual will be normal, whereas 2 individuals are carrier. So 50% of the individuals are carrier, and 1 individual out of 4 will be sickle cell anemia, means 25% of the offspring produced will be sickle cell anemia individual. So the question is, what are the chances of this couple having an affected offspring? Affected offspring means individual with the sickle cell anemia disease. So out of 4, 1 individual will be sickle cell anemia, means 25% of the individual produced will be sickle cell anemia. So the answer is C. Correct answer is C. So that is regarding the first question. Now we will discuss regarding the second question, and the second question is, a man and a woman who both appears normal have a child together who has sickle cell anemia. Sickle cell anemia is an autosomal recessive trait. The woman becomes pregnant again and is told that she is carrying fraternal twins. What is the probability that both of the couple's twins will be developed sickle cell anemia? So here the question is, both man and woman appear normal. So phenotypically they are normal. But the first child produced by them are sickle cell anemia. Sickle cell anemia means with the genotype HBS. So both the homologous chromosome carry this abnormal gene or gene for sickle cell anemia. Now that mother is pregnant again and it is told that she is carrying fraternal twins. Fraternal twin means two different egg fertilized to produce two different zygote. So she is carrying fraternal twins. The question is, what is the probability that both of the couple's twins will develop sickle cell anemia? So what is the probability that both the child of twins will develop sickle cell anemia? That is a question. So from the question we got to know that the first child produced is sickle cell anemia. Means both the chromosome of homologous pair carry the abnormal gene. So out of this, one is received from the father, another chromosome is received from the mother. So from that you can conclude that both the parent carry this abnormal gene or gene for sickle cell anemia. So both the parents are carrier for sickle cell anemia. HBS. So both the parents carrying the gene for sickle cell anemia. Then only this parent can produce sickle cell anemia child. So both the parents are carrier. Carrier means they are phenotypically normal but they carry this abnormal gene for the next generation. So as it is in the case of first question. In the first question, the carrier parent produces sickle cell anemia individual. So what is the probability of carrier parent producing sickle cell anemia? That is 1/4. So out of 4 child produced, 1 child will be sickle cell anemia. So probability of producing 1 sickle cell anemia offspring is 1/4. So probability of producing 1 sickle cell anemia individual by the carrier parent is 1/4. The question is, what is the probability of both the child of the twins develop sickle cell anemia? So what is the probability of producing 2 child being sickle cell anemia? That is very simple. So 1/4 into 1/4. That is 1/16. So probability of producing both the child of the twins develop sickle cell anemia is 1/16. So answer is first option A, that is 1/16. So that is regarding the 2 problems related to sickle cell anemia. I hope this video will be useful. If you like this video, please hit the like button and subscribe to my channel. Thank you.